Good morning, everyone. I'm delighted to welcome you all today. Today, we'll be exploring in-process controls for sterile manufacturing and packing. This is a critical area in ensuring product quality and safety. In this presentation, I'll walk you through key concepts, best practices, and regulatory considerations. So, let's get started and dive into the world of sterile manufacturing controls. Let's talk about real-time monitoring and verification during sterile manufacturing. This is a critical aspect because it ensures that the critical process parameters, or CPPs, stay within the validated limits we've established. By doing this, we're actively controlling the manufacturing process to maintain quality and consistency. Additionally, it helps us confirm that the critical quality attributes, or CQAs, are being maintained throughout production. This continuous oversight is vital to ensure the final product meets all quality specifications. Another key benefit is the prevention of contamination, batch failures, and even the loss of sterility. Catching issues early means fewer costly mistakes and safer products for patients. All of this ties into our broader approach, being an integral part of the quality by design framework and our contamination control strategy. Finally, this introduces the concept of in-process controls, or IPCs, which are the tools and measures we use to implement real-time monitoring and verification. These controls are essential for maintaining the highest standards in sterile manufacturing. Sterile products are considered high-risk dose forms, which means that even tiny levels of contamination can have serious consequences. Because of this, there's a zero-tolerance policy for contamination in sterile manufacturing. Ensuring sterility isn't just about one step, it involves multiple layers of assurance. First, robust process controls are essential. They help us maintain consistency and prevent contamination before it can occur. Second, cleanroom and environmental controls are critical. They create a controlled environment, minimizing the risk of airborne or surface contaminants. Third, equipment integrity and validated systems ensure that our machinery performs reliably and doesn't introduce risks. And finally, personnel aseptic behavior is key. Our staff must follow strict procedures to prevent contamination from human sources. All of these elements work together through an effective IPC, or in-process control, system. IPC ensures that our processes are consistent, that our products remain sterile and safe, and that we stay compliant with regulatory standards. In the context of sterile manufacturing, understanding and controlling these factors is vital. They form the backbone of ensuring product safety, quality, and ultimately, patient health. Let's take a closer look at some key regulatory frameworks shaping our industry today. Starting with the EU GMP Annex 1 from 2022, it emphasizes the importance of mandatory contamination control strategies, or CCS, along with continuous verification processes. It also highlights the need for an effective environmental monitoring strategy to ensure ongoing compliance and product safety. Moving over to the FDA's aseptic processing guidance, the focus is on critical aspects like in-process controls, or IPC, as well as the use of closed systems and maintaining data integrity. These elements are essential for preventing contamination and ensuring reliable, traceable data throughout manufacturing. Then we have the PIC-S Annex 1, which aims to harmonize global expectations. Here, regulators are particularly focused on risk-based in-process controls, meaning that controls are tailored based on the actual risk to product quality. Real-time monitoring of critical parameters is also emphasized, allowing for immediate detection of deviations. Container closure integrity remains a top priority, as it directly impacts product sterility. And finally, the importance of digital, auditable, traceable IPC records cannot be overstated. These records support transparency, accountability, and compliance across the entire process. Overall, these regulations underscore a common theme: a move towards more proactive, real-time, and data-driven approaches to aseptic processing and contamination control, which aligns with our overarching goal of ensuring product quality and patient safety. Now, let's take a closer look at the raw material stage. First, we need to ensure the water quality, whether it's water for injection or purified water, meets strict standards. That means checking bioburden and endotoxin levels to prevent contamination early on. Next, we verify filter integrity both before and after use to make sure our filtration system is reliable and effective. We also perform pH, osmolality, and conductivity checks to confirm the water's chemical properties are within acceptable ranges. Additionally, controlling temperature and mixing conditions during this stage is crucial to maintain consistency and prevent any microbial growth. Moving on to solution preparation, it's important to verify mixing time, speed, and uniformity. Proper mixing ensures that all components are evenly distributed, which is vital for product quality. We also conduct in-process testing, such as checking clarity, pH, and performing assays to confirm the solution's correctness at each step. Setting up filtration properly is another key point. Any errors here can compromise the entire process. Lastly, we monitor hold times carefully to prevent microbial growth, making sure that the solutions stay within safe limits until further processing. Throughout both stages, in-process controls, or IPC, play a vital role. They help us confirm that each step is performed correctly and consistently, ensuring the final product's safety and efficacy. This comprehensive approach, from raw material to solution preparation, is essential to maintain the highest quality standards in our manufacturing process. Now, let's discuss sterile filtration and the key controls involved. First, we use sterilizing grade 0.22 micron filters, which are essential for removing all microorganisms and ensuring the product remains sterile. These filters must comply with PUPSIT, meaning we perform pre-use and post-use integrity tests to confirm their effectiveness before and after use. Monitoring is critical here, so we keep an eye on differential pressure, flow rate, and filtration time. These parameters help us quickly identify any issues like filter clogging or failures during the process. Additionally, the filter validation and certification status are carefully tracked to ensure ongoing compliance and performance. Moving on to equipment IPC, or in-process controls, we monitor line performance, alarms, and fail-safe checks to maintain a robust system. This includes overseeing CIP and SIP cycle monitoring and validation, which are crucial for cleaning and sterilization processes, ensuring they are effective each cycle. We also pay close attention to tunnel depyrogenation temperature and dwell time, which are vital for eliminating pyrogens and ensuring product safety. All these controls are supported by automated digital logging of CPPs, critical process parameters, so we have a reliable record of all activities. Altogether, these sterile filtration and equipment controls form a comprehensive system that guarantees product integrity, compliance, and safety throughout the manufacturing process. Let's talk about the controls in the aseptic zone, which are critical for maintaining a sterile environment. First, we have grade A and B air quality maintenance, ensuring that the air quality consistently meets strict standards to prevent contamination. We also utilize real-time particle monitoring, so we can immediately detect any particles that might compromise the environment. In addition, airflow patterns and pressure differentials are regularly checked to confirm that air flows correctly and that contaminants are kept out. Operator interventions are tracked carefully to ensure proper procedures are followed and to identify any potential issues quickly. Moving on to environmental monitoring, or M, we have continuous monitoring systems in place. These systems track non-viable particles, like those 0.5 and 5 micrometers in size, as well as viable airborne contamination, which includes actual microorganisms. To complement this, we also use settle plates, contact plates, and glove prints to catch any contamination that might settle or come into contact with surfaces. And importantly, we have alert systems set up to trigger immediate corrective actions whenever contamination levels exceed acceptable limits. All of these controls and monitoring strategies are essential parts of infection prevention and control, or IPC, in aseptic processing areas. They work together to ensure that the environment remains as sterile as possible, protecting both the product and the patient. Now, let's move on to component preparation. First, we perform a visual check of the washed and depyrogenated vials to ensure there are no defects or contaminants. Next, we conduct tunnel temperature mapping to verify that the sterilization process is consistent throughout the tunnel. It's also crucial to monitor washer vacuum and pressure controls, as these parameters directly impact the cleanliness and sterility of the components. We then verify the sterility of stoppers and closures, making sure they meet our strict standards before use. Moving into the filling process controls, we focus on fill volume accuracy and pump precision to guarantee each dose is correct. Proper stopper alignment, plunger insertion, and seal placement are essential to maintain integrity and prevent contamination. We continuously monitor rejection rates and analyze trends to identify any process deviations early. Just before sealing, we perform an integrity check to ensure the container is properly sealed and sterile. Throughout all these steps, we emphasize the importance of in-process controls during filling and component preparation. These measures help us maintain product quality, ensure patient safety, and uphold our commitment to manufacturing excellence. Now, let's talk about the critical controls in our process, starting with shelf temperature and chamber pressure. Maintaining precise temperature and pressure conditions is essential to ensure product quality and consistency throughout the lyophilization cycle. Next, we have ice nucleation strategies, such as ATD and ControLyo. These methods help us control the freezing process more predictably, reducing variability and improving the overall efficiency of the cycle. For endpoint monitoring, we rely on instruments like the Pirani gauge and capacitance manometer. These tools allow us to accurately determine when the sublimation process is complete, ensuring the product is properly dried before moving on. Stopper position confirmation is also vital. It helps us verify that stoppers are correctly placed and secure, preventing contamination or product loss. Moving into sealing and packaging, crimp integrity verification ensures that the containers are sealed tightly and securely, which is crucial for maintaining sterility and product stability. We also implement automated visual inspection systems, often AI-based, where applicable. These inspections help us identify any defects or anomalies in the packaging early, ensuring only quality products reach the end user. Container closure integrity testing, or CCIT, involves methods like helium leak testing, HVLD, and vacuum decay. These tests confirm that the container's seal is intact and that the product is protected from external contaminants. Label accuracy and reconciliation are essential to ensure that each package is correctly labeled with the right information, which is critical for traceability and regulatory compliance. Finally, if applicable, we perform in-process controls during lyophilization and packaging. These controls help us monitor and validate each step, ensuring the entire process meets our quality standards and regulatory requirements. Now, let's take a look at some of the rapid microbial methods, or RMMs, that are transforming our approach to sterility assurance. Techniques like ATP bioluminescence allow us to quickly detect microbial contamination by measuring light emitted from ATP, giving us near real-time results. Flow and solid phase cytometry enable us to analyze cells rapidly and with high sensitivity, which is crucial for timely decision-making. Additionally, PCR-based rapid detection methods provide highly specific and fast identification of microbial DNA, significantly reducing detection times. These technologies collectively enable us to achieve near real-time sterility assurance, which is a game-changer for maintaining product quality and compliance. Moving on, digital and AI-driven IPC systems are also making a big impact. Real-time analytics dashboards allow us to visualize process data instantly, helping us spot trends or deviations early. Predictive deviation alerts use AI algorithms to anticipate issues before they happen, so we can act proactively. AI is also being used for particle detection and fill volume control, enhancing precision and reducing waste. Moreover, integrating manufacturing execution systems, or MES, with electronic batch records, or EBR, provides complete traceability from raw materials through to finished products. Now, let's explore the advanced tools enhancing our IPC capabilities. Process Analytical Technology, or PAT, includes inline spectroscopy techniques like NIR and Raman, which enable us to monitor critical quality attributes continuously without sampling. Automated fill volume sensors ensure accuracy during filling processes, reducing variances. Single-use closed systems, along with isolators and RABS, restricted access barriers, are also transforming our manufacturing environments. They help minimize contamination risks and improve overall process control. All these modern technologies are working together to strengthen our IPC systems, making them more robust, responsive, and reliable, ultimately ensuring higher quality and safety for our products. Let's dive into some of the key monitoring points during lyophilization. First, shelf temperature monitoring is crucial because it ensures that the product is being frozen and dried at the correct temperatures, which directly impacts the quality of the final product. Consistent temperature control helps us achieve uniform drying and prevents issues like melting or incomplete sublimation. Next, chamber pressure control is equally important. Maintaining the right pressure inside the chamber influences the sublimation rate and overall cycle efficiency. Precise pressure regulation allows us to optimize the drying process and avoid product loss or collapse. Ice nucleation control, especially with tools like ATD or ControLyo, is a game changer. By controlling where and when ice forms, we can promote uniform nucleation, which leads to better product consistency and shorter cycle times. Endpoint monitoring is another critical aspect. We typically compare Pirani gauges and capacitance manometers to determine when the drying process is complete. Using the right measurement technique ensures we don't under- or over-dry the product, which is vital for stability. Verifying the stopper position might seem simple, but it's essential. Ensuring stoppers are properly positioned prevents contamination and guarantees the integrity of the sealed product. Lastly, if applicable, we also monitor the integrity of the IPC, or inter-process communication, during lyophilization. This helps us ensure that all systems are functioning correctly and that the process is running smoothly from start to finish. All these points tie back to our overarching goal: achieving a reliable, efficient, and high-quality lyophilization process. Proper monitoring at each step ensures product consistency and safety, which is ultimately what we're striving for. Let's take a closer look at some of the key risk-based tools we use to ensure quality and compliance. First, we have QBD principles, which focus on designing quality into the process from the start, rather than relying solely on in-product testing. This proactive approach helps us identify and mitigate risks early on. Next, tools like FMEA and HACCP are essential for systematically analyzing potential failure modes and critical control points, allowing us to prevent issues before they occur. We also leverage trending data, CPV, and deviation management to monitor process performance in real time and respond swiftly to anomalies. Alarm rationalization and escalation plans are vital for ensuring that alerts are meaningful and that the right people are notified promptly when issues arise. But of course, implementing these tools isn't without its challenges. One major challenge is the high dependence on automated systems, which requires us to ensure these systems are robust and reliable. Operator behavior in aseptic environments also plays a critical role. Human factors can introduce variability, so training and adherence to protocols are key. Data integrity and sensor calibration are ongoing concerns. If data isn't accurate or sensors aren't properly calibrated, our risk assessments could be compromised. Additionally, false positives in real-time environmental monitoring can lead to unnecessary disruptions, so we need to optimize our alarm settings carefully. Finally, PUPSIT complexities. Those are the environmental testing requirements for sterile products. Add another layer of complexity to our risk management efforts. Overall, managing these challenges requires a comprehensive and proactive approach, integrating these risk-based tools to maintain the highest standards of quality and safety in our processes. For further support, please contact us. We're here to help with any questions or additional information you might need. Don't hesitate to reach out. Your success is our priority. Thank you for your time and attention today.